Hey everybody, so today we're going to do a quick overview of our activity on the Introduction to Coastal Sticky Notes, which is a basic overview of us looking at different mechanisms of evolution with a specific focus on genetic drift, gene flow, and mutations. So in this activity, you are part of a year-long research expedition to a chain of islands known to have extremely unique flora and fauna, and while visiting the largest island in the chain, Yates Island, you discover a population of coastal sticky notes. This population lives exclusively on the sandy beaches along the southern coast of Yates Island. So let's chat a bit about our coastal sticky notes. Okay, now keep in mind that in this activity, while our species of interest is called coastal sticky notes, some of you may be using tokens in lieu of sticky notes just to save on paper. Okay, now a bit about our coastal sticky notes. Ploidy. Each coastal sticky note is haploid. Okay, so that means that each individual carries only one copy of each chromosome, meaning the total number of alleles in the population is actually equal to the total number of individuals in the population. Keep in mind, this is unique to this situation, right? Usually we have two copies of two alleles for each gene. This population is unique in that they are haploid, right? So when we look at the phenotypes, we have red sticky notes, yellow, blue, and green. And the corresponding alleles for each of these is R for red, Y for yellow, B for blue, and G for green. All right, so there are 4 color phenotypes, and each determined by the sticky note coastal color gene, okay? Each individual sticky note only has one allele for this gene. Now, the reproduction of these guys is also somewhat special. These guys reproduce asexually via budding, right? So new sticky notes split off from the body of their parent and have the same genotype and phenotype as their parent. So if we have a blue sticky note and a red sticky note, then when they reproduce, the next generation, if we started with one red and one blue, we would end with 2 red and 2 blue because the red made one more red and that blue made one more blue, okay? Now, strengths. The strengths of these sticky notes is that they have very long lifespans, so each coastal sticky note can reproduce for many generations. And they also have no natural predators, okay? So nothing will predate on them, and they will be living unless something in your directions tells you otherwise, okay? Now, they do have one key weakness, and that is that they are weak swimmers. So they can swim a little bit, but they are unable to make long journeys into the ocean due to strong currents. So if we're looking at our sticky notes situated on 2 different islands, they cannot freely cross from one island to the other unless the water level somehow lowers to allow for that transition to happen, okay? Okay, so that's our sticky notes. Let's now get started with this activity. For this activity, we're going to start with a starting population of 8 coastal sticky notes, 2 red, 2 yellow, 2 blue, and 2 green. And they're all going to start together on Yates Island, okay? Now, before moving further, go ahead and record the allele frequencies of each allele in the population, right? So right now we are starting with 4 different alleles. We have the R alleles, the Y alleles, B, and G, right? We have 2 of each allele present at this current time. So if we wanted to do a little math, that would mean that we have 0.25 is the allele frequency for each of our alleles on Yates Island as we start this process, okay? Now, with that in mind, go ahead now and answer the discussion questions that are on your sheet or on Canvas, depending on what modality you're using to complete this activity. Now, once you're done with those questions, we're going to move on to part 2, looking at genetic drift, specifically the founder effect. So, for the first time in recorded history, there is a perfect interaction of tides and sediment moving to expose a sandbar connecting Yates Island to the nearby Laurie Island. So several sticky notes explore the sandbar looking for food and move to Laurie Island. When the tide shifts again, the sandbar disappears and the sticky notes are now trapped. The ones that moved are now trapped on Laurie Island, and the ones that were on Yates are now still on Yates Island, okay? So to facilitate this scenario, go ahead and randomly move 2 to 3 sticky notes from Yates Island to Laurie Island, and then answer the discussion questions that are provided for you on Canvas with your lab group. Now, make sure that as you're recording your allele frequencies for each type of allele, you're recording this in a data table so you can track these changes in allele frequencies over time, because at the end of this activity, you're actually going to visualize this data via a graph, okay? Okay, did you know the difference in allele frequency between the original Yates Island population and the new Laurie Island population is known as the founder effect, which is an example of genetic drift, or the impact of chance, random chance, on allele frequencies. The founder effect occurs when a new population is started by a small number of colonizing organisms, which may or may not be representative of their original population in terms of allele frequencies, as you'll see in your activity here. So now it is time for the two sticky note populations, the one on Lori and the one on Yates, to reproduce. So first, place a mark, for example, the number 1, on all of your sticky notes in the same generation to distinguish them from their offspring, right? So we're calling all these guys generation 1. Next, each sticky note produces one offspring, okay? And remember that sticky notes reproduce asexually, so each offspring has the same genotype and phenotype as their parent. So for each sticky note in both of your populations, add another sticky note of the same color. Now, that's not all though, right? Every time we have the opportunity to duplicate our DNA and make a whole other organism, there's an opportunity for mutations to happen, right? So this brings us to part 3, and this is going to carry through throughout the rest of this activity, where every time we have a round of reproduction, there's an opportunity for mutations. So part 3. Every time one of your sticky notes reproduces, there is a chance for a mutation to occur. So for every generation of sticky notes, there's a 25% chance that one of the newly produced sticky notes will experience a mutation. Note that this rate is higher than mutation rates in populations of real organisms, okay? So each time your sticky notes reproduce, you will roll a 20-sided mutation dice. If you roll a 1 to 15, there's no mutations in this generation of sticky notes. If you roll a 16 to 20, pick one random sticky note to experience a mutation, okay? And then you're going to mark that mutated sticky note with the letter M, okay? And a unique number. So all descendants of a mutated sticky note will have that mutation. And all future offspring of mutant sticky notes should be marked with the mutant symbol, since the mutated allele is passed on to offspring, okay? So note that even if the mutation was neutral, this mutation is now still a new allele in your population, right? So for each mutated sticky note, so if you get a mutation amongst your sticky notes, for each of your mutated sticky notes, you will roll a 20-sided mutation dice. If you roll a 1, the mutation is what we call deleterious or damaging, and this sticky note is not able to reproduce moving forward. Okay? If you roll between a 2 and a 19, the mutation is what we call non-functional and has no effect on the sticky note. But if you roll a 20, the mutation is advantageous or useful, and this sticky note reproduces twice as much as they would have normally. So mark this sticky note and all of its future offspring with a plus symbol so that we can keep track of who has double the number of offspring every time they reproduce. And then after going through this first round of reproduction, go ahead and answer the discussion questions that are found on Canvas. Next, after that, we have part 4, looking at gene flow. So one of the purposes of your research expedition is to study the impacts of human activity on organisms living on Yates Island. So for example, records indicate that climate change has influenced the strength of the seasonal tides that pass between Yates Island and Laurie Island. And on very rare occasions, the tides are so subdued that a small number of sticky notes are able to swim between the two islands. So step 1, migration. Choose 5 or fewer sticky notes to move between Yates Island and Laurie Island. You can have them all move from Yates to Laurie, all from Laurie to Yates, or move some in either direction, okay? And then after you do that, go ahead and answer the corresponding discussion questions found on Canvas. After this, you'll do step 2, which is reproduce, right? So you're gonna follow that same reproduction strategy that we brought up the first time, including the opportunity for mutations to arise. All right. Then after that, random chance, okay? So to replicate the ongoing effects of genetic drift, aka random chance, elect one group member to close their eyes and remove 8 sticky notes at random from the two populations. So you should remove 8 notes total between the two populations, okay? And these are sticky notes that perished or died due to random chance before they were able to reproduce, okay? And then after this, go ahead and have all your sticky notes that are still living reproduce again, okay? Again, still following all those rules pertaining to the chance of mutations to occur. And then random chance again, choose another group member to close their eyes and remove 15 random sticky notes from the two populations, okay? Again, you should remove 15 notes this time total between the two populations. These are sticky notes again that died due to the impacts of genetic drift, random chance before they were able to reproduce. And now with this population, go ahead and have them reproduce, right? And make sure that every round of reproduction, you're keeping track of allele frequencies for each generation, all right? So make sure you're filling in that data table as you go. And then we have record and discuss. So after doing all of these rounds of replication and following the directions for gene flow and genetic drift, go ahead and answer the discussion questions pertaining to this section found on Canvas. And then after that, we're going to move on to part 5, which is looking at genetic drift, specifically at population bottlenecks, okay? So disaster strikes. Go ahead and decide with your lab group what kind of natural disaster befalls your coastal sticky notes. Choose one member of your group to act as this disaster, and then this person should remove all of the sticky notes except for 10. So remove all but 10 sticky notes according to the type of disaster you choose, okay? These 10 survivors can be split evenly or unevenly between Yates and Laurie Island, and then have them reproduce, right? Have the remaining sticky notes reproduce, follow the reference documents along with how we handle mutations, just as we have before, and make sure that you are continually, like I said before, recording allele frequencies and their changes over time, generation to generation. After this, go ahead and answer those discussion questions. Did you know this is a simulation of a population bottleneck? A population bottleneck is a sudden dramatic reduction in the size of a population. Often, the allele frequency of the population before the bottleneck is different from the allele frequency after the bottleneck, since the small number of individuals that survive the bottleneck may not be representative of the population's original allele frequency. Okay, well, next week or at your next lab, we're going to build off of this activity and look at diploid organisms that are undergoing natural selection and sexual selection. But for now, this concludes our activity for our coastal sticky notes looking at genetic drift, gene flow, and mutations. So go ahead and take all the data that you've collected thus far, looking at those allele frequencies generation by generation, and turn them into a visual graph. Best of luck.